Okay, I've been instructed to start, and I have two mics, one so you can hear me and one so they can record me, if you're wondering. It's always a pleasure to have an excuse to come back to Los Angeles. I used to work for 6 years just down the road, and have moved around a few times, most recently a year ago to Berkeley. So thank you for coming in after lunch. I'm going to give you a few pop quizzes like this. Actually, this is a really big screen, so it does a disservice to my slide. Can you actually tell what's in the slide? You're not supposed to on this one, but I guess you can make it out. This one is more obvious. So on a normal size screen, the first one looks more like stained glass. But in any case, you probably still don't know what this is, right? You can like have some sort of a guess. There's some codons on there, triplets of nucleotides, right? Right, so these triplets are, of course, what encode the amino acids. And the little circle there, I didn't have a pointer. So right, the little circle here shows you, for example, for this amino acid, you can encode it using a T, and then one of these 4 nucleotides, and then a C. So there's a redundancy in the encoding. And what's actually being shown here is something related to what Aran talked about PCA, which is a way to do a linear embedding of things. This is a sort of fancy modern day machine learning nonlinear version of that called a variational autoencoder. And so just as in PCA, you can reduce things down to some smaller set of dimensions. Here, this is a simple example of taking what are these discrete things, these codons, and having a numeric, a real valued representation of them. And in particular, it's a 2-dimensional representation. So as in, you may have seen PCA many times, this is like 1 dimension, this is the other dimension, and it's a probabilistic embedding. So at any point here, you see a codon, and that's the most likely codon that corresponds to the real values at those 2 dimensions. And the intensity corresponds to its probability. So, okay, great. It's kind of pretty. So that's why I put it in the beginning of my talk. It turns out that there are also these kinds of models, and this way of thinking about DNA and codons is also very useful in protein molecule design, which is something I've become very interested in over the past year or two. And so more generally, though, of course, like evolution has created this very informative structure in our genome. And so for many years, I was a statistical geneticist looking at that. And then I started working a bit in CRISPR guide design, and now more generally at protein engineering. And I'll tell you a little bit about each of those 2 topics during this talk. So they're kind of 2 decoupled stories, although they sound a little bit related. And the first one is work that's mostly done, and it's out in a series of papers over the past few years. And the second part of this is very much work in progress, and if you have any feedback, I would welcome hearing that from you, either during the talk or afterwards. And I think everything I'm going to say is in one of these 2 preprints, one of which just came out last week, I think. Okay, great. So CRISPR, probably everyone by now has heard of CRISPR. And so it actually, started making its way through the media, first in the at some point when it became quite popular in these very famous scientific journals like Nature, things like this. And then later on, it made its way into the popular press like The Economist and the New York Times and CNN. And then, I don't know, who saw this cover of Wired? Every word on here is actually a description of what they think about CRISPR. This is now a few years old, so it apparently says that CRISPR means no hunger, no pollution, no disease, and the end of life as we know it. So it sort of sounds like it's like machine learning. It's going to solve all of our world's problems. And so why not work on the intersection of 2 very hyped up areas of machine learning and CRISPR? So this is actually not my motivation for doing it. It turned out to be a bit ridiculous, though. So after Wired published that, these were a sort of a backlash on Twitter emerged where people were making fun of that Wired cover, I think rightfully so. I'm sure there are comparable ones for various subdomains of AI. And so, but even though it is a very hyped up field, just like machine learning, it really is changing the world, and really stands to reason it will change it much more. And we already have very promising translational results for CRISPR. And so this is actually an out-of-date slide. You can see the years there. I haven't updated it in a while, but there's many already promising translational results about correcting genetic defects in various things from like cystic fibrosis to the receptor that's generally used by HIV to get in. And who here's heard of a gene drive? So gene drive is like sort of man-made creation of super inheritance that can wipe out, say, malaria mosquitoes and possibly other creepy things. I do not work on that, but there's some interesting science there. So, right, but what I am going to tell you about is how I worked with some collaborators at the Broad Institute on the intersection of machine learning and gene editing. And usually when you hear gene editing, you think of the conventional edit of change like an A to a T. But another actually very, very commonly used task is to knock out a gene. Like just prevent it from functioning. And so you can imagine you may do this to query a system to understand like network connections, what happened if I knock this out. You may look at it for drug screens and things like this. This is actually a real workhorse of modern day molecular biology, both in academia and outside. And so there are many things you can do with this, and I'm not going to touch on most of them. I'm just going to talk about two kind of core elements to this system, and that is the sort of on-target problem and the off-target problem. So what I here is a schematic of the genome, and so imagine this is the whole genome, and this little segment here is one gene. And the idea is that I want to use this CRISPR system to prevent this gene from functioning, and I'm going to do that by essentially making a cut in it and then letting it repair itself, but repair itself badly. There's some stochasticity here. If it repairs itself badly enough, then this gene will not function, and this is actually how you leverage this technology. So now the issue is that you can actually cut it in many places in the gene. There's a few constraints of the system. They're pretty mild, so in general I can cut it in hundreds of places, and some of these will work well, and some of them will not work well. And some genes may have many that work well, and some genes may have very few that work well. And right now, it's sort of it has been largely a trial and error endeavor that's lengthy and time-consuming. So of course, if you can have some sort of model that yields a tool that people can use, this will very much help the world. And there were a few ones out there when we started this work, and now a lot of people also use the stuff I'm going to tell you about. But let me also tell you now the second problem. The second problem is this off-target effect. So imagine I've decided that I am going to cut it. Of the hundreds of positions on this gene, I've decided I'm going to cut it right here. Then given that I've cut it here, now I need to be worried about, are there accidental effects elsewhere in the genome? So although it's a fairly precise system, and I'll tell you a little bit more about how that precision works, you can easily have accidents, and you can imagine this wreaks havoc, especially if you're doing this in a living system. If you're just using this for drug screens, it's not sort of as dangerous, but it certainly adds noise to your measurements and makes it harder for you to infer what you would like to infer. Please stop me if something is not clear, by the way. Okay, so obviously there's two paths you can take to improve this. One is that the protocols for doing these things in the laboratory and the actual objects you're using is rapidly changing. But of course, I'm here going to tell you about how we can also use machine learning to do this. And so I don't need to have this in the talk, but I think it's a really fascinating history that I love to tell. So, and it was alluded to in one of the earlier talks, so that is part of an immune system. So in fact, for many years, people looked at these weird repeats in the genome, and they didn't know what it was. And it was not until 2007 that actually a yogurt company, Danone, a researcher there, or some researchers there, figured out by sort of pursuing a problem what it was. And the problem was that some of their vats of yogurt were getting infected and others were not. And so it turns out this is related to an immune system. But what's really incredible is you can see the short timeline from 2007 to 2012 when this thing that was identified as a naturally occurring immune system became co-opted to do general purpose gene editing in many organisms. That's pretty incredible speed of technology. So how does this thing work in its native habitat before we use it for gene editing? So this is a schematic of a virus, of a bacteria that's being invaded by a virus. And this red bit here is supposed to be the viral DNA, if you will. And so what happens is a lot like our adaptive immune system, which is that if the bacteria is able to fight off the virus, then it can take sort of like a snapshot or a memory of that DNA. And the way it stores is it basically puts it in its own DNA. And I'm abusing the terms RNA and DNA here. I'm just going to say DNA because it's easier. So what it does is it takes a bit of the invading RNA and it puts it in here, and it has this sort of scrapbook of viral memories. And now if the bacteria gets invaded again by a virus with that little bit, it's kind of primed to see it, just as our adaptive immune systems are primed to see it. And so that's and what the name CRISPR comes from actually is, it's called clustered, wow, I've suddenly forgotten it, clustered interspaced regular palindromic repeats, something like that. I think it's on the last slide. Clustered regularly interspaced short palindromic repeats. And what that is, it's actually the little bits of DNA that delineate in between these kind of memories so that it knows what's its own DNA and what is not. So that's where the name comes from. And so what people really quickly identified was the fact that there's a precise cutting mechanism here that was briefly described earlier, where you have some sort of a guide that takes these genetic scissors, Cas9, and it guides them to the part of the genome where it needs to make a cut. And in the native system, that cut is a defense against the virus, but in this co-opted system, it's where you're going to induce a repair, like an edit, or you're just going to let it edit itself or repair itself and hope that it does so badly and you knock out the gene. So that's how it works in the system. But what you can imagine is that there's many properties about the guide here that may determine whether it works well. And that if you had a bunch of examples of this, you could maybe use machine learning to predict, supposing I want to knock down this gene, where should I knock it down? And also not knock things down elsewhere. So that's sort of, in a nutshell, how we think about this problem from a machine learning perspective. And the first problem, I would say, is almost kind of commodity machine learning in terms of methodology right now. So it's sort of a straightforward supervised predictive modeling. And what that means is we're going to get a bunch of examples. So the tricky part here is actually getting these data and working with really brilliant molecular biologists who are clever and know how to do that. And it's actually quite difficult in many cases. And I'll tell you a little bit about that in just a second. But in terms of the machine learning, you basically want a bunch of examples where you know the sequence of that guide. These are just a bunch of nucleotides, roughly 30 nucleotides long. And then you're going to get measurements as to how effective it was at measuring the gene, and you want to estimate some function that will work for examples you haven't seen and tell you this. And so then in future, someone says, I want to knock down this other gene that you were not trained with, then hopefully this gives you a good answer. You have some rank order of things to try, and you can be more efficient and more cost effective than you would have otherwise. So I'm not going to say too much about the machine learning here. There's some interesting sort of scientific things that come out if you probe at the features that turn out to be useful. One example here are the thermodynamic features of the guide. What you can see here is the double-stranded DNA. This big thing here are the scissors. That's the Cas9 that are going to come in and make the cut. And as you can imagine, as you can see, it has to pull apart the double-stranded DNA. And this depends a lot on the thermodynamics, which in turn depends on the sequence properties. And so this is the kind of thing that even if you don't necessarily know about that, if you have enough data, machine learning can automatically extract. Of course, we actually don't have a lot of data. And to the extent we have someone who gives us insight into which features might be good, this turns out to be very useful. We're nowhere near the kind of sizes of data that you see which are breaking benchmarks in speech and audio. So, and we could have used a neural network here, and this might have been on the cover of Science because it was a CRISPR and neural networks, but they did not perform better than more conventional models, so we didn't do that. So I'll just say very quickly that although that was kind of mundane machine learning methodology, there's some, as often happens when you start to tackle just a problem with a regular methodology, you uncover more interesting problems. And you can come talk to me offline about this if you're interested. But one thing that's very complicated in this data that I glossed over is that the supervised label is actually measured in systematically different ways for different kinds of genes. So in some cases, it depends on that gene being expressed on the surface of the cell and being tagged with a fluorescent marker. In other cases, depends on a priori known drug sensitivity. And so in some sense, you have these supervised labels that don't live in a coherent measurement space. And so this is an opportunity to develop new models that automatically kind of align those spaces jointly as you're doing the supervised machine learning. And so this is not a problem we've solved, but we think we're about halfway there. If you have something that matches that template, I'd be interested in talking to you. And so anyway, after a lot of due diligence and careful thought and testing, we ended up with, at the time, I don't know if it's still true, a model that outperformed the other models out there and became used throughout the world. I'm not sure if it still is. Is anyone here using it? Okay. Right, so that's the on-target problem. And so we didn't want to stop there because we knew the off-target problem was just as important. And so I'm not going to say too much about this off-target problem, but from a methodological and statistical standpoint, it's much more difficult. And there's basically two reasons that it's more difficult. The first is that if I'm considering that I'm using this guide, but I want to know if there are accidents elsewhere, then just sort of logistically, I need to scan across something like 3 billion nucleotides. And so this is just a large feat to scan it for a single guide. And now if I want to do this for multiple guides, it just becomes very expensive. But perhaps more interesting from a statistical standpoint, I need to look for now accidents where the guide and the target are mismatched. And they can be actually mismatched up to, say, 6 mismatches. And this poses a large statistical power challenge because we don't have nearly enough data to warrant that kind of combinatorial complexity. And so we had to sort of do some little tricks there to get it working. I'm not going to talk about them today in the interest of time. But now the sort of modeling is slightly different because now the guide and the target are not by definition matched. In fact, by definition, they're kind of not matched. And so you need to sort of also tweak the modeling in that way. So I'm not going to talk too much, or I'm not going to actually talk more about this at all. I'll just say that by the time we did the on-target and the off-target model, the other thing we really wanted to come out of this was a tool that people could use. And so we told our collaborator at the Broad we would like to compute all of these scores for every guide that hits a coding part of the genome. And he laughed at us and said, that's impossible. And so at that time, I worked for Microsoft and I had access to quite large compute. And we said, no, we can do it. And so what we did is it went through, actually, this paper went through 5 versions in the editorial process. 3 of those required a repopulation of this database. Each of those we ran 17,000 cores for a month straight. And I don't think my startup money will cover that. And where are the Amazon people? Who has those academic credits? Okay. So yeah, I could use some. So that's kind of the CRISPR story. And although protein, and this can be used, of course, for protein design, all kinds of stuff, but I'm now going to tell you a separate story, which is protein design, which is actually completely decoupled from that. And I meandered into this almost by accident by chasing some weird project with an intern that over the course of a year kind of morphed into this. Okay, so here's another why slide. Does anyone other than Ben Raphael know the answer? He got it, actually. I showed it to him, and he knew immediately. Someone must know. It's a Princeton effect. It's a pretentious artist in a Paris gallery, and the gallery apparently warned that it could take longer than anticipated. Okay, so that's not really terribly impactful on society, but in fact, searching for like the needle in the haystack is very important in science in many practical applications, and in particular for protein design, where you need to kind of search through a really massive combinatorial space again to find what you're looking for. And so that's the thing I've become very interested in recently. And I started this for proteins. It turns out that the chemists are also very interested in this, and there's a mapping for this problem that applies to both of these quite naturally. So one example, for example, David Baker's lab at University of Washington has done a lot of work on protein stability and trying to create de novo proteins that can still fold, which up until they did this was basically impossible. And so, but roughly speaking, when I'm talking about search, and I'll get a bit more precise in a second, I'm saying that there's many sequences we can consider, and a huge number. I'll show you in a second how many. And we in some sense want to search efficiently through this space of sequences to find the ones that satisfy the properties we care about. There may be one or more properties. You can imagine you want to say maximize binding to some target, subject to that it stably folds and things like this. Sometimes this is referred to as like multi-objective optimization in drug development. So this is maybe the one connection to CRISPR. So you could imagine doing this kind of thing actually for Cas9, and people have started to get at this, is like how could you actually make it bind more efficiently? So that's just one example. And there are many things people want to do with proteins. You want to make them fluoresce more. And I mentioned that there's connections to chemistry also. I'm not going to say more about that. But so how do the combinatorics look here? So imagine, this is back of the envelope calculation here with no constraints on a particular protein. But if you go just from 5 amino acids, how many sequences with that alphabet can you get? Get a pretty large number. If you go to just 20, you already are at a number of sequences that's larger than the number of grains of sand in the world. And remarkable, I don't know if this is a conspiracy theory, but the last time I put this slide up, the speaker before me had this exact same number, and he said it was the number of seconds that had elapsed since the Big Bang. I don't know where we're getting these numbers, but maybe they're approximately correct. It's quite fascinating. So the point here is that I think machine learning can help us to look through this search space better, and in particular better than what people have been doing so far. So let me formalize that a little bit more. So this is the problem we've been thinking about. So we're going to assume, for one, that we've kind of, we don't want to have to interact with a lab. We want this to all be in silico. Of course, you could do better if you can also augment with laboratory measurements, but this is a separate problem that we're not yet addressing. So for first things first, we're going to assume that we have what we call it an oracle, is a predictive model for some property we care about. And for now, I'm just going to assume I care about one property because it's simpler to tell the story. And so in particular, I'm going to assume that I have some, maybe it's a neural network, maybe it could be anything, just some black box that gives me a DNA sequence and then tells me how much I get of the thing I care about. And then I may want to maximize that, for example, right? So I want a method that will tell me what sequence to choose to, which DNA sequence to choose to, say, maximize the protein yield, or we have a sort of specification version of the problem in which I want to say, I don't want to maximize it, I want a particular value, which is a sort of very similar problem. And then, of course, we want to have constraints in here. If I just do this in general, this is not super useful, right? So I may want to say the DNA must actually code for this exact amino acid sequence. And there's still a combinatorial explosion here because of the redundancy I showed you of the codon vocabulary. Or I may actually be willing to change the amino acids, but I may say you have to adhere to certain structure elements so that I know, for example, you're going to function according to these things I already know about. So you may have some set of constraints as well. And so the goal, and then finally I should say, is we want this to be what we, in computer science, I don't know exactly who's in the field, call this a black box in the sense that I do not want to depend on knowing the internals of this thing. And in particular, I don't want to depend on doing like computations like gradients on it. So it's just an input to output mapping. And the reason I want to have only that dependence is that there's so many scientific domains where there are expert models that have already been developed, and we just want to be able to grab them and plug and play with them and not have to like retrain models that are suitable for our method. So I think the problem is clear, right? I've been given this input to output mapping, and in some sense I want to like specify the output and go and get the input. So, and I'll tell you a little more carefully about how we do that. But just before I get there, let me tell you how this is done now for protein engineering. And it was kind of, so I actually knew Frances Arnold was doing this kind of stuff, and I was going to go approach her to see if it was possible to collaborate, and then she won the Nobel Prize, and then I figured I'd have to wait 10 years before I could get a response, although I've since been in touch with her students. But so she got this Nobel Prize this year for something called directed evolution, and it's actually a very simple but obviously incredibly high impact method. So how does it work? Imagine you want to make a protein, say, fluoresce more, which is something people do, and it's kind of a canonical system for these things. You start off with some parent strain, and maybe it has a bit of the property you care about, and then you go through a diversification step. So that may mean that you subject it to, say, X-rays, which induce random mutations, and now you have a variant pool of that, with just random mutations that somehow differ. And then what you do is you need some laboratory procedure to measure the thing you care about, so maybe fluorescence, for example, and you pick off the top performers. And so this is the sense in which you're directing evolution. So you're giving it the fitness function, this is the property you care about, and you're directing it because you pick off only the top performers, and then you repeat the cycle. And so, right, so what we would like to do is, one, replace the laboratory measurement with a predictive model, although that's not the really interesting part. It's just something we require to do the rest. And the thing that we really want to do is do something that's smarter at looking through this huge search space, right? So if you think about what directed evolution is doing, it's inducing just random mutations, and then it's like doing a greedy kind of walk through the random mutations. So there's no way that this is an effective way to navigate this huge combinatorial search space. Okay, so how are we going to think about this? So although I said we want a black box, I've drawn a neural network here, which is not a black box, but just because it makes it easier to talk about. So right, a normal predictive model, and this is what we're going to kind of call our stochastic oracle, is that you give it a bunch of inputs and a bunch of outputs, as for example in the CRISPR work that I told you about, and then you change all these knob settings, right, these W's, such that it predicts the things you care about well. And what we're going to do is we're going to essentially turn that on its head. We want to be able to say, given such a network that already has its parameter settings, we want to specify some number of properties, either their value or to maximize them, and then find the sequence that matches these. And so on the face of it, this is, if you know combinatorial optimization, this is kind of just that problem, but it starts to look different when you get into the problem a little bit more. So one reason that it's a little bit different is that if you're a good machine learning person, you will consider uncertainty of your model, although this is not necessarily very in vogue at the moment. And you actually should care about how certain it is that it's about its predictions, and also whether these are calibrated uncertainties. That is, like, does the uncertainty have some sort of meaning? And so we want models where the uncertainty has meaning, and we want to also leverage this in our walk through the space. So places of the space that are more uncertain, we should think about differently than places that are certain, for example. So, and all of this work is, I was really lucky when I started at Berkeley a year ago I got my dream PhD student as my first student, and so we've been working on this over the past year. Everything I'm going to tell you about is joint work with him. And so I don't want to get too much in the technical details, but I'll give you a little bit of a flavor of the technical aspect of this. So how are we going to solve this problem? So remember that we've been given this sort of oracle, this probabilistic oracle that goes from input to output, and a probability distribution because we care about uncertainty. And what we're going to do is we're going to use a generative model. So probably people here may not be familiar with that term. A generative model is some model that generates objects that we care about. So in this case, it's going to generate sequences, like protein sequences, and hopefully it's going to generate sequences that satisfy the property we care about. And the generative model, so you could think of, for example, a Gaussian distribution. It has a mean and a variance. And when those are set, it tends to generate things that have that mean and variance. If I change the knobs, the parameters, which are the mean and variance, now it starts to generate different things. So we're not going to use Gaussians. We're going to use some much more sort of richer classes of models that generate vastly more complex objects. But similarly, they have knobs like a mean and a variance. These we call theta. And as you change this knob, it tends to generate different sequences. So now the game we're going to play is, can we change the knob such that it generates the sequences we care about? So it sounds a little bit magical, but it's just sort of part of the formalism of how we tackle this problem. So that's one thing we have, is a class of generative models that has a knob, and we're going to turn that knob. And then the other thing that I've said numerous times is we have this sort of stochastic oracle, which tells us probabilistically if the sequence under consideration satisfies each of the properties and with some uncertainty distribution. And as I said, the game is going to be to find the setting knob phi at iteration t when we're done that's going to yield our desired protein sequence. And you might be asking, well, it's going to generate many, isn't it? Well, so in fact, if your oracle is uncertain, then you don't want to end up with one solution because you actually have uncertainty that's inherent in the system. So this is actually a property that you want to emerge from such a solution. So I'm not going to go into the technical aspects of this. I'm going to just skip over this, but I would just want to emphasize, because there's similar work out there where they just kind of pull this out of a magic hat and similar things, and it doesn't work. And this one actually works, and it starts from first principles, and you can actually derive the solution that we've arrived at. But it has a very pleasing, intuitive, algorithmic outcome, and so this I will tell you about. And moreover, it maps kind of quite nicely to directed evolution. So the procedure is that roughly you need this oracle I told you about. This is its mean, and this is essentially takes account for the uncertainty, the CDF, but just think of this as that oracle object. So I said we're going to assume we've been given that. We're assume we have some sort of a generative model, and it can take examples, sequences, each of which are weighted. So the higher the weight, the more it affects the kind of sequences that the trained generative model will give off. And we need a procedure that will train it with these samples. And we can optionally also take some, how much does this adhere to the property I care about, and with what kind of certainty? And based on what it tells us, then we will weight that example appropriately. Those that adhere more to the property we care about will get a higher weight when we train the generative model, and correspondingly those that don't will get a lower one. And you just repeat this as you would in directed evolution, and we can talk about stopping criteria offline. But at the end of the day, what you end up with is a generative model, like whose parameters have slowly shifted, such that this distribution is yielding protein sequences of the kind you care about. And it has some, you can dig into the math a bit under certain circumstances. For example, if that predictive model it has 100% certainty, then under some conditions, the generative model will collapse to only giving you one sequence. And that's actually the kind of behavior you want. And to the extent that that oracle is uncertain, it will not do that. It cannot do that. So I think that's quite a pleasing, intuitive result. And so we developed this kind of in a small bubble, not knowing who was connected to anything else. And we've been talking a lot about it. And so people have been telling us now, and now we've learned quite a bit, how this is actually related to reinforcement learning in some ways, not necessarily in terms of the goals that are stated, but in terms of the statistical and machine learning machinery that's involved in solving this problem. it has a resemblance to it. And so we've been reading a lot of this, and I think there's going to be an interplay between these areas, very surprisingly. And it also has strong ties to image generation, which has become a very popular field right now, although and so in particular, this paper here actually has a lot of similarities to some of the things that I've just told you about. And so, right, so it's early days for us. I really want to do this in an end-to-end sort of wet lab validation, but there's a few sort of sweet spot biological domains where this will be most useful. And as you can imagine, one of those is where you have a predictive model, or you have the ability to build a reasonably good predictive model. And the other is where you can construct the sequences that we tell you to construct. So for example, we were talking to George Church's lab in Boston, and they said, yeah, it would be great to do that for CRISPR, but CRISPR's so big now, it would be really hard for us to produce those things. And so it's too early stage. So we're right now kind of talking to a lot of people working in protein engineering and in chemistry to see where we can do this end to end. So what I'm going to show you right now are simulations that are just meant to sort of validate the way we're thinking about this is correct. And they're also based on real data, so that the simulated data is somehow projected into a space that's realistic and that we care about. And so in this first example here, this is based on simulated data from a paper that came out earlier this year by one of my colleagues at Berkeley in Nature Biotech. It's a really incredible paper, rich with data on gene expression, fitness, I think it might even have ribosomal footprinting, all kinds of stuff. And so we basically trained a neural network on that and then pretended that that was the ground truth and then did the rest of the simulation from there. And we compared it to other methods out there. And so in particular, I said, there's this was done from first principles. This model right here was kind of, it's very, very similar. If you read it, the algorithm is almost identical, but there was no sort of rational basis for how they did it. And it turns out to perform substantially worse for this problem. And you can see in this example, it's a pretty short example in the sense that we have only 96 nucleotides, but already we're at a quadrillion sequences. So there's no way we can enumerate these and take a look at them. So there's some sort of technical jargon up here. This is under the assumption of an unbiased oracle. What does that mean? Basically, that means that we're assuming the oracle is correct. Noisy, but in expectation it's correct, and that's the behavior. And then, so something that's really fascinating is to look at how you can not just maximize, but actually specify how much of something you want. And that's important for things like protein stoichiometry, where you have sub sort of units of a protein, and you need to have them in exactly the right ratio for it to fold and then be active in the way that you want it to be. And you can't just create them separately and mix them in the right ratio. They actually have to do that all at once, sort of in one mix, if you will. So this is also something we care about. And again, this is another example, and it just shows basically that we can do it. We're not, other people actually were not really trying to do this, so we're not comparing here. But one thing I'll highlight from this that's quite interesting is this variable here. This is the noise of that oracle. So this is how certain is that predictive model of the property. And very pleasingly, intuitively, when the oracle is less certain, that's the blue, it has a higher variance, it gives you a larger distribution in the solution. That's because it's more uncertain, so that's exactly what you want. And when the noise goes down, it gets a tighter distribution. So again, this is like a very intuitive behavior that you hope would emerge. So this was the first preprint we put out in around October, and since then we now decided that it's not quite right. And the reason it's not quite right is that you're never going to get an unbiased oracle, but we learned a lot by doing that, and I think it's actually still a really beautiful method. But in a real world, this predictive model we build and its uncertainty, it is going to be wrong. And in particular, it's going to be wrong outside of where it's been trained. So if you ask almost any machine learning person if they believe that extrapolation is possible on a real data set, they will say basically no, it's not. Maybe you can push the envelope a bit. And so what we decided was that we would like to push the envelope as much as we can. And how can we balance sort of the predictive model and where it's confident with also knowledge of what's sort of a reasonable state to be in. And so the second version we have is a very related method, but where we now sort of, we give it a set of examples that we state are reasonable. So for example, it's known which proteins fold. So you can imagine giving it a list of like hundreds or thousands of proteins that are known to fold, and you say you must somehow adhere in some very vague, non-specified way that like to the properties of this very large set. But given that, now go do what you were doing before. So somehow you're incorporating prior information to not let it go too far into the extrapolation space and stay reasonable. And so we think this is the one that's actually going to be very useful in practice. And so I think, what time is it? Oh, I have 3 whole minutes. Okay, so great. So this is actually a rather sophisticated simulation because we needed to be in a setting where we knew the ground truth and we had a biased oracle also. And what that means is that it emerges quite naturally. If you just train a model on a certain domain and then you go a bit outside of it, it actually just does badly. And it's a bit of a complicated plot, but let's see if I can explain it a bit. So our method, well, let's look at one of the worst methods. This RWR is actually a method that some reviewers told us to compare to from robotics. It's the link, if you will, to reinforcement learning in robotics. And it's particularly susceptible to going crazy if the oracle has any errors in it. And so what you see is as the iterations go, we're trying to do maximization of a particular property here, the fluorescence based on a real data set. And as time goes on, this is what RWR thinks is going on, because this is what the oracle's saying your fluorescence is getting better and better. But the training data actually only only went up to about 3.1. So already really closely in the beginning, we're well outside of the domain of the training data. And the ground truth is actually the solid one. And we know because we constructed this. And we didn't game this. We just sort of trained these models. And so actually, the outside, it's going way, way down. And that's, you see these plots all the time about if you have a linear trend, like what does it do after? Maybe it's going down. And so in a lot of these models, it does. You see these weird things. And so in some sense, the reason you want this prior knowledge is to stop it from hallucinating in this way. And so in contrast, our method here, and the way it does this again, is with this sort of set of prior knowledge proteins. Ours says it thinks this is the oracle. And it actually, if you can see it, it's actually below the ground truth in our case, and this is the ground truth. But the important thing is that they have sort of a constant width between them. They're tracking each other. And so this is what ends up keeping it into like a well-behaved part of the space. Okay, so I think maybe I spoke too quickly about too many things, but I will stop there. And thank you for listening.